बिस्मिल्लाहिर्रहमानिर्रहीम। शुरू अल्लाह के नाम से जो बड़ा मेहरबान निहायत रहम वाला है। As we all know that this paper is for 1 hour and this is the alternative to practical. This used to be called paper 6 but now it is called paper 4. So we have two variants, 4.1 and 4.2. Now let's look at the first variant, and this is the November 23 exam. Hydrogen peroxide is a toxic waste product found in cells. Catalase, an enzyme present in cells, breaks down hydrogen peroxide to produce water and oxygen. So catalase enzyme is present in cells, and we can take potato cells or we can take liver cells, any plant or animal cells, and they all contain catalase. So this breaks down hydrogen peroxide to produce water and oxygen. And oxygen is the one which we're going to measure because it's going to produce some sort of bubbles. When potato tissue is added to a solution of hydrogen peroxide in a test tube, the catalase in the potato cells, please understand, catalase in the potato cells causes bubbles of oxygen to be produced. If some detergent is added to the hydrogen peroxide solution, the oxygen bubbles are trapped to produce a layer of bubbles that rises up the test tube. The height of this layer of bubbles indicates how much catalase activity there has been. A student used this method to investigate the activity of catalase in potato tissue by following these instructions. Label 3 test tubes A, B, and C. So we have 3 test tubes A, B, and C. Add 5 cm of hydrogen peroxide solution to each test tube. 5 centimeter cube. Please look at the volume as measured in cm cube. Hydrogen peroxide is an irritant, may cause damage to the eyes and skin, so you should wear goggles or anything which is eye protection, goggles, and wear gloves so that when you handle this. Add 1 centimeter cube of the detergent. So then we add 1 centimeter cube of the detergent. We have added 1 centimeter cube, so the volume is going to remain constant. Add 1 centimeter cube of the detergent of hydrogen peroxide in each test tube. Then cut 3 pieces of potato measuring 10 millimeter by 10 millimeter. So we cut a cube 10 into 10 into 10. Add one of the piece of potato in the test tube. So then you add one piece of potato in this test tube, one in this test tube, and one in this test tube. So they all have the same surface area. Add one of the piece of potato to test tube A. It will sink to the bottom of the hydrogen peroxide solution. Immediately start your timer and observe the piece of potato. Bubbles of oxygen will rise to the surface and form a layer at the top of the hydrogen peroxide solution. Measure the height of this layer at 2, 4, and 6 minutes. 2 minutes, then 4 minutes, and then 6 minutes. So you have enough time to do it. From the start and record these measurements. The height of the layer of bubble should be measured from the top of the hydrogen peroxide solution. So from here to here, the top of the hydrogen peroxide solution. From the top and to the top of the layer of the bubble, they're shown in figure 1.1. So they've shown you this in the diagram and they've said, okay, this is what you've got to do. Okay, so then you cut the second piece of potato into 4 equal parts. Okay, so there I was wrong in the beginning. So you take this piece of potato which you had, 10 into 10 into 10. I cut the second potato into 4 equal halves. So you'll cut it like this and then you'll cut it like this. So cut the second piece of potato into 4 equal halves, equal parts, and add them to the test tube. So this 4 parts we are going to add to test tube B. And then start your timer and repeat the measurements as for test tube A. So you repeat that for test tube like you did for test tube A. Cut the third piece of potato into 8 parts. So the third piece of potato you're going to cut into 8 parts. Right? So cut the third piece of potato into 8 equal parts and add them to test tube C. So this is C and this is B. B has 4 parts and C has 8 parts. Right? Start your timer and repeat. State a safety precaution that the student should take while doing this investigation with hydrogen peroxide. Wear goggles, or you can say wear gloves. Any of these was correct, and you could have written this. Wear eye protection, safety spectacles, gloves, use gloves, or wear goggles. Now it says the student recorded the measurements in a notebook as shown in figure 1.2 and enter the headings in table 1.1 and complete table 1.1 by entering the student's results. So this is the figure 1.2. So enter the headings. So then you have to go on to table 1.1 and you have to enter the headings. So what are you going to do here? Here you have time and what is the unit? So time in minutes. And here what do you have? What will be the heading? Height of layer of bubbles. And then you must write the units, which is in millimeter. So you have completed the headings, time in minutes, and height of the layer of bubbles in millimeter. Now you have to complete it. So 2 was, looking at the top, you can see it there. 2 was what? 12, 14, and 18. So you wrote the headings, time in minutes, height of the layer, and then of course you completed it. And this was all for 3 marks. So value in each cell, data entered in correct columns, data entered in the correct line rows. So if you got it all correct, this was, of course, there were 2 marks for this. First the headings, there's 1 mark for this and 1 mark for this, and then for this there were 3 marks. So the 3 marks for mainly was value in each cell. So you wrote something in each of the columns, and then this was correct, and in the correct columns, and then the correct lines. If everything was correct, well, you got 3 marks. Then it says cutting the potato pieces into smaller parts increases the surface area of the potato. Describe the effect of increasing the surface area of the potato and explain what caused this effect. You see, when you had 8 pieces, 6 pieces, in C you had 6 pieces, so there is 19 millimeter, 31 and 38. So in this situation there was more surface area. Now it says in the question, describe the effect of increasing the surface area and explain what caused this effect. So more bubbles produced. So more bubbles produced got you 1 mark for the description. But the explanation was why. You see, when you have this, then you have the enzymes only on this part of the cells which were exposed. But when you cut it up into 4 pieces, then all the sides are being exposed. You have this piece, this piece, this piece. Now you've increased the surface area, and the enzyme is all in the potato here. I mean, it's all over here, around here. So more catalase in contact with hydrogen peroxide. Here we have just 1 piece. Here we have 4 pieces. So more catalase or more enzyme in the potato in contact with the hydrogen peroxide. So more oxygen produced. Please understand it, when we've cut it into smaller pieces, now we have more area which is being exposed to the hydrogen peroxide. Hydrogen peroxide is the substrate. The potato cells contain the enzyme. So more enzyme in contact with the hydrogen peroxide, H2O2, which I've written as this. But you should write the whole thing, hydrogen peroxide. Now coming to the next part, the student found that the top of the layer of bubbles was not always flat when they had to measure the height of the layer. Suggest what they could do to ensure that the measurements taken every 2 minutes were comparable. So you then have to say, okay, if it was, if in the test tube it was something like this, then every time either you measure the middle point. It says always measure to the highest or the lowest or the midpoint of the surface. So every time either you measure this top part or you measure this part, highest, lowest, or you can say the midpoint, which is this part. So if it was something, you see, it could be anything. It could be something, we could have a test tube like this, and the things could be like this. So then highest would be this point, or you say the lowest, or you can say the midpoint. So if every time you're measuring the highest, then that's fine. In all of them, you've done that. So suggest what they could do to ensure all the measurements taken every 2 minutes were comparable. You could compare them and say, okay, well, that's the same. Like for instance, in one test tube you measured this point, and then another test tube you measured this point. So well, then that is not a very comparable comparable readings. So always measure to the highest. Either you said the highest, which is slash, that means either of these was correct, or the lowest, or the midpoint of the surface. Part 6 of the question, it says, it's part, yeah. The student noticed two unexpected problems during the investigation. Suggest how each may have affected the results. Number one, bubbles sometimes stuck to the potato. Suggestion: bubbles would not increase the height measured. Some of the 8 parts of potatoes in test tubes C stuck together. Now this would be what? This would slow the reaction, or the surface area of the catalase not increased as planned. So they said, it says the student noticed two unexpected. Suggest how each may have affected the results. So if the bubbles were sticking to the potato, then that means they did not they did not go and affect the height. So this is the potato here, and then a bubble is sticking here to it. But then the bubbles forms this bubbles on the top, right? These are all the bubbles on the top. So if this bubble sticking here did not go, so it's okay, bubbles would not increase the height measured. So the height you are measuring is this. Now this bubble is not part of that bubble. So it's okay. Suggestion, then some of the 8 parts of potato stuck together. Then in another test tube, we had 8 pieces of potatoes. So the 8 pieces of potato were not separate, and some of them are sticking together. So that means that surface was not being exposed. So this is one piece of potato, and then this is another piece of potato. Now all 4 sides are being exposed. But this one was sticking to it. So this part is not exposed now. So this will result in a slower reaction. And what we had planned, that the number of enzymes would increase because there would be different pieces of potato. So there would be enzymes here and there would be enzymes here. But now because they are sticking to each other, so that the catalase not increased as planned. Part 7, it says, suggest how the student can prove that it is the activity of the enzyme catalase that produces the bubbles. Now, I always give this example. That's a very simple example. If we say uh, a certain student X is making all the noise in the class, and we think that student X is making all the noise. So we say, okay, class with the student X, and then the same class without the student X. So if still there is noise, that means it wasn't student X who was making the noise. It must be student Y or student Z. So number one, what you can do is that you can boil the potato. These potato pieces which you have used, if you boil them, it's going to denature the enzymes. And if you still put them in the test tube and you add the hydrogen peroxide and still bubbles are produced, well, then that means it wasn't the enzyme which was working, it was the bubbles were produced because of some other reaction. But if you have boiled the potatoes, then all the enzymes should have been denatured and no bubbles should be produced. Or what you can do is you can take the potatoes and not boil them, but instead of hydrogen peroxide, you just add distilled water. Now, the water does not contain the substrate. Hydrogen peroxide is the substrate for that enzyme. So water is not going to be the substrate, so no bubbles are going to be produced. So either of this was correct. Either you use distilled water instead of the hydrogen peroxide, or you boil the potato to denature the catalase. So use distilled water instead of hydrogen peroxide and boil the potato to denature enzyme. Then coming to the B part of the question, another student investigated the activity of catalase in potato by measuring the volume of oxygen produced. So we've got hydrogen peroxide here, we've got the potato here, we've got a flask, and then they added the cubes of potato to hydrogen peroxide solution in the apparatus shown in figure 1.3. And then what we've done is we've collected the oxygen. Name the piece of apparatus labeled D. What is it? It's a measuring cylinder. So it's a measuring cylinder. Design an investigation to determine the effect of temperature on the activity of catalase in potato. Use the apparatus shown in figure 1.3. So we have to use this apparatus, and you have to design and investigate, determine the effect of temperature on the activity of catalase in potato. Now, how are we going to do it? Now, some of you one day asked me this question is, can we just do a diagram? Yes, you can do a diagram, but if you know how to do a diagram, that is the main thing. So now I have drawn a diagram like this, and I have tried to show you how you could even do a diagram and you could draw it. Like for instance, it says all the three flasks take at least three temperatures. So you said take 5 flasks or 3 to 5. I always say take 5. So the fact that you take 5 different temperatures or three different temperatures, and then you identify those temperatures 30 degrees Celsius, 35, 40 degrees Celsius, or you can take 45 and 50 as well. Then the use of a water bath. Then you give time for equilibration time, which I've talked many times in many papers, is allow time for the hydrogen peroxide to come to that temperature before you add the potato. In fact, the potato and the hydrogen peroxide should be at the same temperature. So you should have kept them separately so that they achieve the same temperature. Then same volume of hydrogen peroxide. Are you going to add 5 cm cube or 6 cm cube or 7 or 8 or 10? So same volume and same concentration of hydrogen peroxide. Then measuring cylinder full of water each time. This measuring cylinder full of water each time. And then you measure the volume of gas. Then you measure the volume of gas produced in say 1 minute or 2 minutes. And then you compare the volume of oxygen produced at different temperatures. So how much was it at 30, how much was it at 35, and how much was it at 40? Now, as it says, use the apparatus shown in figure 1.3, so you must refer to that. So 5 different temperatures, 25, 30, 35, 40. You're writing the units is very important. Degrees Celsius, use a water bath. Give time for hydrogen peroxide to come to temperature before the potato is added. Same mass or the same volume of the same potato. Then same volume, or if you said the same concentration of hydrogen peroxide, like you say 5 cm cube of 1% hydrogen peroxide. Then the measuring cylinder should be full of water each time, as I showed you here. This is the measuring cylinder here. So you would see that this measuring cylinder D is full of water each time. And then you would measure the volume of gas produced in say 2 minutes. So you say measure the volume of gas produced in stated total times in 2 minutes or 3 minutes, or you say that you denote the time taken to collect 10 cm cube of the oxygen, so a fixed volume, or you say the gas produced in 2 minutes or gas produced in 3 minutes. And then you compare the volume of oxygen produced at different temperatures, and that of course gives you a very good idea. More the volume of oxygen produced, more is it near the optimum. Now I want to explain something to the very brilliant students who will be wanting to try for an A and an A star. Now for instance, I took three temperatures 20, 30 and 40, and I got these 3 points, and here I have the volume of oxygen produced. So here the volume of oxygen produced is say 10 here, and then it's 20 here, and then here it's gone down to something 15. So at 20 I have this value, then at 30 I have this value. Let me just put it a little right. So at 30 I have this value here, it's here. Now you realize if I have these value, and at 40 we have this value here. Now if I connect these points, now just I'm going to show you how there's an error here. So you connect this point and then you connect this point. But say you had more readings between 30 and 40, and you had a point here, and then you had a point here, and then you had a point here. because you took readings between, say, 32, 34, and 36. So look at how the graph is now going to change. I'm going to use another color. The graph is going to go up like this, and then it's going to go down, and then it's going to go down. So first you were thinking that the optimum was 30, but now when you look at it, the optimum is more towards 36, because this is 32, this is 34, this is 36, and this is 38. Now the optimum is somewhere here. So you realize how just taking 3 temperatures was not very exact. You needed to take more temperatures between 30 and 40, because at 30 you see the rate was here, at 40 the rate has decreased. So the optimum is somewhere between 30 and 40. But you didn't take any readings between 30 and 40. Now you realize how this is a very, very, very minute and a very intelligent point for the students to realize that this is not going to be exact. So then, okay, how are you going to write this? I was just explaining this to a student and she didn't know how to write this. So I said, okay. So then you say the optimum is somewhere between 30 and 40. So you take more readings between 30 and 40. So 32, 34, 36, 38 degrees Celsius, and you do the experiment again and see how many bubbles are being produced. And that would give you a more accurate reading. First you were saying 30 is the optimum. Now you're saying 36 or 38 is the optimum. So you get a more accurate reading. When we say reliable, then we say repeat the experiment. R for reliable, R for repeat. That means we've done this experiment 5 times and every time we've got nearly the same reading. Just a matter of a few, maybe a little bit here and there, that's fine. But when you repeat the experiments, then the results are reliable. So when you repeat the experiment, the results are reliable. But when you're doing this 32, 34, 36, 38, then the readings are accurate, because previously you said that the optimum was 30. Now you're saying the optimum is 36. So you realize how we mean, where do we mean to say accuracy and where we mean to say reliability. So that finishes this question 1, and we will continue the next question in the second video, which follows this video. Thank you very much.